Anybody have any questions? I will say a little bit more about the exam on Thursday. I'd like to try to make sure we make it through the material today, because my intent is to stop the material for the first exam today, and then we'll start with new material on Thursday. But there's the breakdown. It's really the same breakdown as the 2 old exams I have posted on the website. I would encourage you to look at those exams as kind of final review. I do make up new exams, so don't just memorize the answers for the old exam questions. But it gives you an idea of the kind of questions that I ask and the length of the exam, etc. Okay? Nothing? Okay. When we talk about co-transport, I really only talked about it in the context of using this hydrogen ion gradient that was created by a hydrogen ion pump to move some hydrogen ions across a membrane and to establish a high concentration on one side of a membrane. Anytime you have a high concentration of something on one side of a membrane, particularly ions, and a low concentration on the other side, that represents potential energy to do work. And in the case of the plant cells, they allow those hydrogen ions to flow back through a channel from high concentration to low concentration, and in the process kind of pull along sucrose with it. That's called sucrose co-transport. Okay? Well, as I said, you can learn something every day. Really, my basic understanding of the sodium-potassium pump was that it was really most responsible, most significant in neurons. And it's very significant in neurons. Because if a neuron's going to fire, when it fires, sodium goes rushing into the cell and potassium comes out. And you've got to reestablish sodium out here and potassium in here so the neuron can fire again. So I knew it was important in neurons. But talking to Dr. Rhodes, it really turns out that sodium ion gradients are really important in most cells. And cells spend a significant amount of energy constantly pumping sodium out of the cytoplasm back into the environment. In effect, as he refers to it, charging the battery to create a potential energy gradient. And then they couple the internal transport of sodium down its gradient with the transport of other key nutrients and things that the cell needs to get. Dr. Morton was just telling me that he did some work. Was this postgraduate?
No.
When you were in life. With this very thing. And so it's very significant and very interesting. So the cell kind of uses a general mechanism to charge this battery to pump ions out here, and then uses that potential energy to power several different transport processes. So sodium, potassium, ATPase pumps are not just important in neurons. Okay. I love computers. We've spent some time up to this point talking about membrane functions, things like transport, okay? Things like potentially cell-cell recognition. But one of the things that you hopefully understand now is that these functions are going to be directly related to the structure of the cell membrane. And we've talked a little bit about the structure, but not in any kind of comprehensive way. So this last section I want to talk about will relate the functions we've talked about into the... Oh, great.
You want to restart.
Yeah, I might want to. Let's see. So to more intentionally make this correlation between membrane structure and membrane function. One of the things we'll be talking about are some of the models of membrane structure that have developed over the years and kind of the history of membrane model development. A model, in reality, is really a hypothesis. Okay? It's a tentative explanation for scientific phenomena. And over the years, the model of membrane structure has changed as new information has been gathered about how membranes behave and what they're made up of. In part, technology has really helped us to gather new information that has allowed these particular models to develop. There we go. So let's head on down here. Our current understanding of membrane structure is that the cell membrane is a fluid mosaic of lipids, proteins, and carbohydrates. And I'll explain in just a second what fluid mosaic refers to. What's important to understand is that this realization really is relatively recent. We'll talk a little bit about the history of the development of membrane models over time, which date back to the 1800s, actually. This most recent model of membrane structure came out about the time that Dr. Morton and I were in grad school, in the 70s. And this has held up pretty well over time. And all of the experimental evidence that's been gathered about membranes is consistent with this hypothesis. But does that mean that sometime down the road this model might be displaced by another model? Absolutely, that's a possibility. That's what happens in science. As things constantly change, ideas change, models change, based upon gathering new information. So we're pretty confident about the fluid mosaic model, but it's not infallible. Okay, this is a brief history. I put dates in here because I like to give you some kind of a context of when these things happened. But I don't expect you to memorize dates, okay? Names, I think, are a little bit more important. I think it's important to understand some of the basic names in science. I don't give a lot of them to you, but I do think when we start talking about experimental evidence and models, it's important to associate people with those models. So, if you go way back to 1875, okay, Civil War time, some of the early evidence about cell membranes was that Overton believed that they were made up of primarily lipid, in fact, exclusively lipid. And this particular hypothesis was based upon an observation that he made. That lipid molecules, or other nonpolar molecules, had an easier time crossing the membrane than polar molecules did. He believed that the membranes were lipids because when substances move across the membrane, it's almost like they're dissolving in the membrane. And this is very consistent with an old chemistry idea that like dissolves like. Okay? Polar solvents tend to easily dissolve polar molecules, but they don't dissolve nonpolar molecules, and vice versa. Okay? So he looked at the movement of molecules across membranes and found that lipid-like molecules moved across more easily than non-lipids. So, therefore, membranes were made of lipids. Fast forward about 40 years, and Langmuir, in 1917, said, I'm going to make a little refinement to this. I think that the lipids are actually a special type of lipid called a phospholipid. Okay? And we talked a little bit about phospholipids. and particularly that phospholipids are these schizophrenic molecules, or these amphiphilic molecules, fancy word for it, that had a hydrophobic and hydrophilic portion. Okay? So they're kind of unique lipids. In 1925, Gorder and Grendel said, we believe that the cell membrane is actually a phospholipid bilayer, 2 layers of lipids. And they're organized together so that the hydrophilic portions are pointing out toward the environment and toward the cytoplasm where there's water in both places. And then this region right here is hydrophobic. But why did they think it was a bilayer? That was actually based upon some physical measurements they did. They were looking at red blood cells. Red blood cells are a great kind of cell to use to study cell membranes. Because basically a red blood cell is a sac of hemoglobin. Okay? Does anybody know a fundamental way that red blood cells differ from all other cells? They have no nucleus. Okay? So they have a limited lifespan. Your red blood cells turn over about every 90 days and die. They don't have a nucleus to code for new proteins. So basically they're a big transport sac for hemoglobin. Okay? Well, they took red blood cells and they measured their surface area. Measured their width, their length. And then they extracted the lipids from the cell membranes. And they tried to figure out whether the proportion of lipids correlated to the surface area of the membrane. And they found out that they had twice as much lipid as they had surface area. Therefore, phospholipid bilayer. In 1935, Davson and Dinelli proposed a model that was kind of a departure. They said, yeah, okay, phospholipid bilayer. But additionally, they proposed proteins being part of this membrane structure. And they envisioned proteins as coating the surface of the membrane on both the exterior surface and the interior surface, giving this kind of sandwich-like appearance. Okay? So the bread was the proteins, and the jelly was the phospholipid bilayer. And this was what was in our books when we were sitting in your point in undergraduate biology classes, the Davson and Dinelli model. There was actually some interesting work done by Robertson in 1950 where he actually did electron microscope pictures of cell membranes and found out that they had this, what he referred to as a trilaminate structure. This one's a little bit hard to see. So here's a cell membrane electron micrograph. Here's the outside of the cell, here's the inside of the cell. And you see this layer, layer, layer. That 3-layered structure, trilaminate. And he interpreted this as being Davson and Dinelli's idea of a protein layer, a phospholipid bilayer, and another protein layer. Okay, so what appeared to be direct visual confirmation of the Davson and Dinelli model. And that's pretty much the way the idea of the cell membrane stayed until 1975. When some information began to be accumulated that was inconsistent with this idea of membrane proteins only being on the outside or the inside. For one thing, that really didn't explain the importance that membrane proteins played in transport. That was a big part of the disconnect. So in 1975, Singer and Nicholson developed this fluid mosaic model of membrane structure. They ended up receiving a Nobel Prize for it. And Larry, wasn't Nicholson actually a postdoc or something? I don't think he was a PI. Yeah. And this represents diagrammatically the fluid mosaic model of protein structure. Now this is one we've seen already. I showed you this earlier on, and we talked about it in the context of kind of communication between outside of the cell and inside. So what does the fluid mosaic model say? Well, it says that the membrane is a phospholipid bilayer. That's no big revelation. But they said that the proteins were not confined to just exterior or interior surfaces, but that the proteins can be distributed both on the surface of the membrane, like some of these maybe on the internal surface, but that there were a number of proteins that span the phospholipid bilayer. Those were called transmembrane proteins. Now, you might say to yourself, well, how is this possible? Because how can this protein have part of its structure exist in this hydrophobic core of the membrane? Wouldn't it be unstable? Wouldn't it hate to be there? Well, remember that proteins are complex chains of amino acids. And amino acids have R groups. Each amino acid has a characteristic R group. And some of those R groups are polar, and some of those R groups are nonpolar. Remember also that these amino acid chains fold up into complicated three-dimensional structures. And if a protein folds up properly, it could fold up so that this part of the polypeptide chain that was exposed to this hydrophobic core would be primarily the nonpolar amino acids. And the polar amino acids would more constitute this interior part of the structure of the protein, which are shielded from this hydrophobic core by their nonpolar cousins. Okay, so you can fold these things up in such a way that everybody's stable, and at the very end I'll show you a picture of what that might look like. They also said that this was not a static structure, that the membrane had fluidity. Well now, based upon the context of what we talked about in terms of membranes function, membranes have to be fluid. Okay? How can you have a static thing like that piece of dialysis tubing you're going to work with in lab and have a vesicle pinch off of that? Or have a vesicle add to that membrane? Or have the membrane pinch in during endocytosis? The membrane's got to be fluid to do that. So what they suggested was these phospholipids are not in fixed positions, but that these phospholipids can move from side to side, from place to place. This phospholipid might be here at this moment. Might be over here the next moment. And so there's a fluidity to this particular membrane. It's really kind of like an ocean sea, okay? And the individual water molecules move around in the sea. Individual phospholipids also move around within this bilayer. Now, what about these proteins? Can they move? Possibly. In fact, many of them do. They kind of bob along almost like little icebergs floating in the sea. And they can move through this phospholipid bilayer from one position to another. But sometimes they don't move. Sometimes they're anchored. And they stay fixed in position. Down here, you've got a membrane protein that's anchored to the cytoskeleton. Up here, we've got an external protein that's anchored to some of these collagen fibers, for example. So some of these membrane proteins can move, but others are fixed in position by attaching to other molecules. Yes?
What are the carbohydrates on the surface for?
What are they for?
Yeah.
Can you hold out for just a second? We'll talk about that.
Yeah.
What was the name of the proteins that you gave that were in the phospholipid bilayer?
That were in the phospholipid bilayer? Well, these transmembrane proteins are the ones that kind of span the membrane. Okay? There are some that are found only on the outer surface, and those are called peripheral proteins. There are some that are found only within the bilayer, and they are called integral proteins. I think those terms are used in the study guide, and so that's what they refer to: periphery on the outside, integral on the inside, and the transmembrane across the whole thing. Okay, this shows you the potential movement of phospholipids in the bilayer. And lateral movement of phospholipids from one place to another occurs very frequently. A typical bacterium is about 1 to 2 micrometers in length. Pretty small. A phospholipid that was at one end of a bacterial cell in like 1/10 of a second can migrate to the other opposite end of the bacterial cell. There's tremendous amount of movement of those phospholipids within the bilayer. What's an environmental factor that would influence the rate of movement of those phospholipids? Temperature, yep. As molecular motion increases, won't the membranes have greater phospholipid movement? And the fluidity of the membrane will increase. What's going to happen to the fluidity of the membrane as temperature decreases? It'll become less fluid, it'll become more rigid. The membrane almost gets kind of frozen up. Is there a relationship between fluidity and permeability? The answer is yes, there is. If membranes become too non-fluid, if they become too frozen up, they tend to lose their permeability. Yes?
I understand that, so I understand like why they need to move, but why would they want or need to move from one end of the...
It's really all random. It's just all really random collision and molecular motion. Okay? So there's no real intent or need. That's just the reality of what they're able to do based upon colliding with one another and sliding around. Okay? Now, what is pretty rare is for a phospholipid to flip from this side of the membrane to this side of the membrane. We call that flip-flop. Why would that be rare? Why would that not be thermodynamically favorable?
Do they have to pass through the hydrophobic region?
Yeah, this hydrophilic head would have to pass through this hydrophobic region. So these molecules on this side are free to move kind of back and forth from side to side very easily, but it's very unusual for this one to flip over here or this one to flip over here. Now, in addition to temperature, there are some other factors that influence membrane fluidity. Membrane fluidity is influenced by the types of phospholipids that make up that bilayer. Now, remember what a phospholipid is. A phospholipid is this polar head bonded to two fatty acids, those little tails. Remember that there are two types of categories of fatty acids. What are they? Saturated and unsaturated, right? And what's the difference between saturated and unsaturated? Double bonds are present in carbon to carbon. In which one? Unsaturated. Okay? When you have carbon to carbon double bonds in unsaturated, these phospholipids aren't linear. They tend to have kinks in them. The double bond forms this kink. So this would represent a membrane with a fair number of unsaturated phospholipids. And note how these kinks kind of prevent this membrane from packing together, from getting too viscous. So this would be a fairly fluid type of membrane, as opposed to this, where you've got membranes associated with a lot of saturated fatty acids, and these can tack together very tightly, and consequently this membrane would be less fluid, it'd be more viscous. So at the same temperature, less fluidity, more fluidity. Okay? Well, think about the challenge that organisms face, particularly plants, when their environment changes and the temperature decreases. Okay? Animals might be able to find shelter. The plants are stuck there. Okay? So what if the temperature starts to drop seasonally to a plant? What's going to be the risk? Its membranes are going to become not fluid. Okay? Molecular motion is going to slow down. Fluidity decreases, the permeability is all messed up, and that can kill the plant cells. But plant cells can respond metabolically to changes in temperature. And they can shift over and they can start making phospholipids that have more unsaturated fatty acids versus saturated fatty acids. Okay? And consequently, as the temperature decreases, this will help this particular membrane to maintain its fluidity and to maintain its permeability. So cells of organisms can respond to environmental changes by adjusting their metabolism and producing different types of molecules. Okay? What if the temperature gets really, really hot? Then the membrane might become too fluid. Okay? And so the cell could adjust again and start producing more phospholipids with saturated fatty acid components. And then they would pack more tightly together, and that would help to minimize that temperature increase and maintain the proper level of fluidity. And then finally, there's cholesterol. Cholesterol gets a bad rap. And high serum cholesterol can be associated with coronary artery disease. We talked about that in the context of the familial hypercholesterolemia. But cholesterol is an important, essential part of animal cell membranes. Cholesterol molecules are interspersed among this phospholipid bilayer. Okay? They're nonpolar molecules. They're happy in here. And cholesterol molecules tend to add some rigidity to the membrane. Okay? And keep it kind of intact. Oops. That's bad spelling. Bacteria typically do not have cholesterol molecules in their cell membranes. Does that mean that cell membranes don't need to be rigid in bacteria? Not as much. Because like plant cells, bacterial cells have a cell wall that helps to compensate for this lack of rigidity in the membrane. There's one type of bacterium, though, that lacks a cell wall. And that is one of the few bacteria that we found that actually has cholesterol in its membranes. And so it's kind of a compensation for it. Well, I don't have a cell wall to help maintain my membrane in a rigid state, so I better have this cholesterol instead to compensate for that. Okay, let's get this right now. One of the ways that a study of cell membrane proteins has really been investigated heavily is through a technique. It's pretty tricky to do. Called freeze fracture. In freeze fracture, what we do is we take plant or animal tissue and we very quickly cool it in something like liquid nitrogen that freezes it immediately. And then you cleave this tissue with a very, very sharp edge knife. And oftentimes these blades are made of diamond. for example. And what that can do is it can fracture and split the cells, and oftentimes you will get a split in such a way that it actually splits open the membrane, and then you can see the two halves of the membrane when you look at it with an electron microscope. Oftentimes the split occurs around these proteins that are part of the membrane structure. So this half of the membrane peels away from this half of the membrane. And when you look at that, it's very common for the internal half of the membrane to look different than the external half of the membrane. That's because there's a different distribution of proteins. There are different proteins associated with this part of the membrane than there are this part of the membrane. The membrane is really a mosaic of proteins. It's all these different proteins kind of patched together within this phospholipid bilayer, and these proteins have a variety of different functions. This is an interesting experiment that illustrates that at least some membrane proteins are able to move within the phospholipid bilayer. In this particular experiment, two different types of cells were isolated, mouse cells and human cells. And membrane proteins were labeled with fluorescent antibodies that would specifically bind to those proteins. And the antibodies have different colors if you look at them under a fluorescence microscope. So if we labeled mouse cells with these red fluorescent antibodies and human cells with these blue ones, what's going to happen to those proteins if we stick these two cells together and trick them into fusing with one another? And there are some tricks you can do. One of the things you can do is add this special virus that tends to cause cells to fuse together. It's called the Sendai virus. So, make these two types of cells with different labels on them, add them together in culture with that virus, and they start to fuse together to form a hybrid cell. After a while, you'll have a single cell. If these membranes were not able to move within the phospholipid bilayer, then you'd expect all the blue ones to be at the bottom of the cell and all the red ones to be at the top of the cell. But after a period of time, these membranes equally distribute themselves, so they're all mixed together within that single cell membrane. So, what are these proteins doing? Well, we've talked about some of this already. Some of the membrane proteins play an important role in transport. They might be these ion channels right here, or they might be these membrane proteins that can swivel from side to side and transport things across the membrane. We've also talked about the fact that some of those membrane proteins have enzymatic activity. The sodium ATPase enzyme, for example, is an enzyme and an ion pump. From the material you read for your quiz, they can be involved in signal transduction. So hormones, for example, could bind to a membrane receptor like a G protein. And when that binding occurs, it sets off that signal transduction pathway that ultimately results in something new happening within the cell. We talked about membranes involved in intracellular joining, things like gap junctions. We mentioned cell-cell recognition, where one cell learns to recognize another cell. Maybe a white blood cell membrane is checking out this cell to see if that cell is really belong there or not. And those white blood cells, like the macrophages, might engulf it if it is foreign and its cell-cell recognition patterns don't match up. And then there's also a lot of roles that these membrane proteins play in attachment to the extracellular matrix and the cytoskeleton as well. So Jesse, I think you asked the question about what this stuff was doing out here.
The carbohydrates?
Yeah. Carbohydrates play a number of roles, and one of the roles that they play is in this cell-cell recognition phenomenon. So a lot of these molecules that are on the surface of the cell are actually glycoproteins, and those sugar molecules are a particularly important thing that the receptors on these cells recognize. and determine whether these really belong or not. Some of the glycoproteins are also involved in this translation of information about what happens out here and then stimulating a response in here. So it really is a way for this particular cell to react with other cells and also to get signals from its environment. This is the last figure I said that I was going to refer to right here. This is a pretty complicated protein that is folded up into a complicated three-dimensional structure. And it's got several regions of these helical structures. And in this particular case, several of these little bundles right here all orient themselves into kind of a tube. So in the middle, there is this hydrophilic channel, and then all of these little bundles right here of secondary structure are associated with the hydrophobic core of the membrane. So a lot of non-polar amino acids are found on these external surfaces of this little tubule. So you can have a lot of folding of the polypeptide chain to create these various channel proteins which function in transport. Okay, good place to stop. Don't take off yet. This will give me an opportunity right now to say a little bit about exams since I finished a little bit early. As I've been making the exam, here's what I've been doing. I've been pulling up the PowerPoint slides. And I've been going through the PowerPoint slides and saying, okay, what on here is worthy of an exam question? So my first advice to you would be is to sit down and start reading through those PowerPoint slides with the accompanying notes that you've taken. Because many times they're just diagrams, okay? While you're going through that, I think it would be very helpful to start going through, if you haven't already, reading through the study guide questions, because they tend to follow the sequence of the PowerPoint slides, and it forces you to kind of write down answers to those particular questions. That's the kind of thing you can do and should be doing now on your own to get comfortable with it. What about kind of last-minute tune-up, like a night or two before the exam? Do not wait to do the review of the notes until a night or two before the exam. That is when you could be sitting down with someone in the class, if you wanted to, and talking through this stuff and asking each other answers to study guide questions, for example, or even posing your own questions. If you can explain this to someone else, you understand it. If you are just trying to memorize my words, then you haven't completely internalized it. If you can teach it, you know it. Okay? So at the end of your studying, it really is a good idea to sit down and talk to one another, and you learn as much by explaining an answer to someone else many times as you do by hearing the explanation. The old exams are the last thing that I would look at. That's kind of a final tune-up and say, okay, if I'm properly prepared, I should be able to answer virtually all of these questions. Okay? Now, there might be occasionally some question on the exam where I just haven't covered it that much this semester. There are certain things I tend to emphasize one semester and not so much another. So if you see something that just looks totally foreign to you, we probably didn't cover it. Okay? I am not going to pull things out of the book we have not talked about in class. So you should consider the textbook to be kind of a resource. If you're having trouble with something that are in the notes or the PowerPoints, then go to that chapter, particularly look at those figures and diagrams in there. And they should help you to reason through it. And if you can't, you can talk to me, go to a mentoring session, but it's not too early now to begin studying. Okay? So please take advantage of the resources on the website, the study guides, the old exams, and start looking at your lecture notes. That's really most important. Did I see a hand coming up here someplace? Question? Hey, John.
Is the section on the properties of water going to be on there?
The section on the properties of water that Dr. Morton went over with you, yes, it will be on there. Okay, so all of the study guides and the PowerPoints that are posted, water is chapter 2, and water is mainly, polar nature of water, what physical properties are the result of polarity, and pH, okay? And then chapter 3 was mainly functional groups, some discussion about the properties of carbon. At the very end, a little discussion about dehydration synthesis versus hydrolysis. That's important. Functional groups, you should be able to recognize them, okay? I won't ask you to draw functional groups, but if you see a particular functional group, you should be able to tell me what it is. And they're the same ones that are on those PowerPoint slides. So that's chapter 3. Chapter 4 is cell structure. Chapter 5 is membranes. So basically those 4 chapters. But the majority of the questions come from 4 and 5 because that's what we spent most of the time with. Okay. I have put this up there for the last several days. My hope is that someone would be interested enough in following up on this, because there is a real dilemma there. So if you happen to follow up on this and you want to gain some bonus points, these are not optional, but bonus, then if you could spend the next few minutes writing down what you found, maybe even going so far as trying to communicate to me that you understand a little bit about how the cell accomplishes this kind of dilemma, then you can do it. If you haven't followed up on that, then obviously you can't contribute to this, and you're free to go. So if anybody followed up on this and wants to stay around for a couple minutes and give me a little feedback, turn it in, and you could get some bonus points. Okay? Tomorrow we're going to start on cell energetics, or Thursday. And so you should start reading the next chapter, chapter 6. Energetics is conceptually difficult. So please make sure that you pay attention carefully in class on Thursday, because it's not intuitive.
Hey, I have a question. My mom wants to try and get me Thursday to go home for this weekend for family stuff, but I have cell bio. I was wondering if I could, on Friday, I was wondering if we were doing anything important Friday that I can't miss, right?
No. No, we always do something important, but no, what you can do, I realize things like this come up, is that I'll be recording the lecture, so really what you could do is you could sit down with your PowerPoint slides and the lecture and basically catch up what you would have missed.
Okay, I always print out the notes.
Yeah, there's no quiz or anything that day. There's nothing that you'll have to turn in, so.
All right, do you want me to email you?
Sure, yeah, just to remind me, yeah. Yep, if that's okay. I know that sometimes families things like this.
Thank you.
7th, which is the Friday before.
We are not taking a test. No, the tests are on the syllabus. Test days.
Yeah, but I'll be gone from.
Okay. That's not unusual. There's something we will be doing in class, but it is something that you can do on your own time.
Okay.
So remind me about that.
Okay.
Lab procedure.
Oh, yeah, just bring that to lab.
Okay, just the lab? All right.
Yes.
Do you want me to make that now?
You can.
I don't have another class.
Sure, why don't you just go next door and I'll bring it down?
Okay.
I guess that's all.
Okay.
About that.
And that's all I have to say about that.
He was here,
Who's that?
Rashawn.
That didn't go
Okay.
through one row.
It didn't go through one row?
Yeah.
Okay. So is he the only one you see on there?
Gabby's here.
Pardon me?
Gabby's here.
Yeah.
Yeah, Rashawn was here. He was sitting over
Yeah,
back.
yeah, I remember seeing him. So it looks like everybody else is. Oh, Seth? See Seth?
I'm not sure who he is.